Basel, Switzerland, 1951. The young biologist Willy Bergdorfer publishes his doctoral dissertation under the guidance of Dr. Rudolf Geige. Rudolf Geige was a chemical engineer and biologist whose company produced the red dye in the Nazi flag. During his research, Bergdorfer studied African ticks and the bacteria contained within them. The process of separating the hemolymph from the tick was cumbersome and involved essentially performing surgery on the tick, but he invented a new method, which was to pull off the leg of the tick in order to have the hemolymph flow out. During this research, he became an expert on the spirochete bacteria Borrelia. Hamilton, Montana, 1951. Bergdorfer travels to the Rocky Mountain Laboratory after his PhD in order to study the Rocky Mountain spotted fever. This fever was severe and caused huge issues for settlers in the 19th century. So the Rocky Mountain Laboratory was set up essentially as tents and single-room buildings at first in order to study the fever. And at this point they've gotten an expert from Switzerland in their lab. Bergdorfer and his peers curated one of the largest collections of tick and tick bacteria in the world, and particular focuses of their work was on Borrelia and Rickettsia bacteria. During this work, a challenge was the mass production of the bacteria that they wanted to study. What they came up with was a method of inoculating chicken eggs with the diseases that they want to study, and they would have racks of hundreds of eggs inoculated with these deadly bacteria. Suffield Experimental Station, Alberta, Canada, 1953. Bergdorfer attends a field test of sarin nerve gas organized by Fort Detrick. Fort Detrick was the increasingly large United States chemical and biological weapons program. With expansive resources, connections to Canada and the UK, and Bergdorfer being an expert on what was then a very serious disease was a prime candidate for biological weapons research. The U.S. Army Chemical Corps says, "The advantages of arthropods, which includes ticks, as biological weapons carriers are these. They inject the agent directly into the body so that a mask is no protection to a soldier, and they will remain alive for some time, keeping an area constantly dangerous." This was the beginning of Bergdorfer's research in collaboration with Fort Detrick. This is specifically a military project. They were interested in using ticks as a method of delivery of deadly diseases as a biological weapon. Tick-borne illnesses are complex because on one hand you have many different species of ticks, which each have their own geographic ranges that they live in. And you also have a variety of dangerous bacteria and viruses that inhabit the ticks. Some of the bacteria will kill each other, and sometimes you can pack multiple diseases into one single individual tick. And there are many bacteria that are unclassified. And in addition, many of the diseases caused by ticks are going to be a consequence of multiple bacteria interactions, which makes diagnosis of the sickness very difficult. In order to build a more effective weapon, Bergdorfer develops a method of force-feeding the ticks in the image you see here, with vials prepared with the bacteria and viruses that you want to study. So at this point in the Rocky Mountain lab, Bergdorfer is essentially in charge of huge production of ticks and the production of diseases in those chicken eggs. One of the bacteria that they studied was the Asiatic relapsing fever, Borrelia gladychevi. At this time period, the United States military was conducting tests such as Operation Big Itch in 1954, where they gathered up over 500,000 infected fleas into a plane and dropped them onto an area full of cages with guinea pigs. And then they went to the area and measured what percentage of the guinea pigs were infected from the disease contained in the fleas. Another experiment that they were doing at this time was releasing ticks into the wild in order to detect how quickly they spread out. And they tracked them by dosing the ticks with isotopes, carbon-14 and iodine-125, in order to follow the ticks, essentially with Geiger counters. The United States was interested in using a weapon essentially exactly like this against Cuba as part of Operation Mongoose. Eventually, Operation Mongoose culminated with the Bay of Pigs invasion of Cuba. The goal of the biological weapons research, as far as an insect-borne disease for Operation Mongoose, was to deliver a disease to sugarcane field workers and to get them sick in a way that doesn't kill them but makes them too sick or weak to continue working in the sugarcane fields, which at that time in Cuba's history was essentially their major industry. Their idea was that if Cuba can no longer export sugarcane, then by some fantastical means the regime is going to collapse. During this program, the Rocky Mountain Laboratory was pulling a bunch of separate pieces together in its work on biological weapons. Bergdorfer was at the center of all of this. The Biological Subcommittee Munitions Advisory Group, which is under Fort Detrick, writes in 1966, the use of multiple agents to achieve prolonged incapacitation was also being investigated. In this time period, Bergdorfer went to London to study virology and tick-based virology. One thing he learned was transmission electron microscope analysis of microbes, which gives you a lot more information about any individual bacteria, And in addition, he learned a method to detect antibodies, which is based on using a glass knife to cut thin samples of a specimen and studying it with either the transmission electron microscope or ultraviolet light. Antibodies are signatures on a bacteria which an immune system or a diagnostic test could identify. So detecting antibodies is very valuable if you're studying medicine or biological weapons. At this time period, the military had discovered new methods of microbe growth based on an artificial substrate instead of chicken eggs, and a new method of storage based on freeze drying. This meant that they could mass produce biological weapons and then freeze dry them to transport them to the location that they want to attack. Microbe genetics was the #1 priority. In addition, the Rocky Mountain Lab was investigating specifically Rickettsia rickettsii as a weapon. The method of developing bacteria that Bergdorfer used was essentially breeding. You put the bacteria into a tick, you get the tick to lay eggs and reproduce, and then you infect a bunch of livestock with those ticks, and the ones that fared the worst, you take the tick back and then extract the bacteria, grow that culture, and then repeat the process. The same as how a dog breeder breeds dogs. Bergdorfer, in an interview in 2013, Bergdorfer admits that he was specifically attempting to identify ways of obscuring the antigens. This would be valuable for a biological weapon, because if your biological weapon doesn't have an antigen, it can bypass an individual's entire immune system, and it can't be diagnosed very easily. So, suppose you could develop a weapon based on 3 components. A sickness caused by a tick-borne bacterial contagion, which means an already existing disease isolated, and then augment that with a microbe that masks the antigens of the sickness. And in addition, another bacteria that causes indefinite relapses. The original sickness could be any common disease. An agent that masks the antigens would have to be something very particular, but this was what Bergdorfer was researching at the time. And something that causes indefinite relapses could be very similar to the Asiatic relapsing fever, which Bergdorfer was aware of. This could be grown using the new techniques from the military as a biological weapon. At the same time as this research was being conducted, there are a few unusual tick-borne disease outbreaks in the period from 1968 to 1976. The earliest in 1968 on this graph is the Rocky Mountain spotted fever, which was localized on the west before then. Babesiosis is a common disease associated with ticks, but in combination with other diseases it can be very severe. Lyme disease appears in 1972 in the first major outbreak in history. These newly occurring diseases were shooting up exponentially, and a lot of people were worried because they tended to avoid easy diagnosis. According to this news article, these cases of Rocky Mountain spotted fever were suddenly increasing in the Northeast, but they weren't increasing in Montana, so. Later on, Willy Bergdorfer receives international fame for his discovery of the cause of Lyme disease, and the bacteria Borrelia burgdorferi is named after him. Lyme disease avoids easy diagnostic testing, so this was like considered a pretty major success in medicine, and that's what he's known for today. While he was doing this research, he separately discovered that the sera of Lyme patients also reacts with the antigens of another bacteria called the Swiss agent, which he was aware of after researching that bacteria in Switzerland. Bergdorfer himself suspected that the Swiss agent had a serious connection with Lyme disease itself. Essentially, he suspected that there was an American version of the Swiss agent, which he thought that because the Lyme patients appeared to have antigens that were similar to the Swiss agent and not similar to any other obvious choice. And the Swiss agent was known to have relatives in the US, and in 2016 they figured out that Rickettsia helvetica itself is present in US tick. Despite all of that, the Swiss agent was not mentioned in research after April 1978. This picture is Borrelia burgdorferi, which causes Lyme disease. Let us imagine the following hypothesis. The 1972 outbreak of Lyme disease, the first in history, could be the release of a Fort Detrick biological weapon developed on Bergdorfer's shoulders with components that I described before. Lyme disease as the underlying sickness, a bacteria developed from the Swiss agent as an antigen masking agent, and the addition of some type of relapsing fever or a variant thereof to cause the relapsing features present in a condition called post-treatment Lyme disease syndrome. Well, this is one hypothesis. Would this have been possible for Fort Detrick to develop before 1972? Well, Borrelia burgdorferi was present. Rickettsia helvetica was known, and they would have needed to develop this or augment it in a way that masks the antigens of Lyme disease. And for the addition of some sort of microbe that causes relapsing features, you would need to develop that in the same way that they were developing other biological weapons. Well, is it impossible? I have no evidence to believe that this is impossible. In an interview during the end of his life, Burgdorfer answers in the affirmative to the question of whether the Lyme-infected ticks that he studied after the outbreak contained pathogens similar to those he studied as early as 1952. If Borrelia burgdorferi was the only cause of Lyme disease, he wouldn't have answered in the affirmative to this question. What that means is he was aware of at least some additional factor in Lyme disease related to his own research, which Fort Detrick was keenly aware of. What remains to be understood in this hypothesis is how exactly did such a biological weapon travel from Hamilton, Montana, or wherever it was ultimately developed, to Lyme, Connecticut. I'll present three theories. Theory number one. The Soviet Union mobilized its existing network of spies within the Manhattan Project to infiltrate Fort Detrick chemical and biological weapons. After succeeding in exfiltrating active specimens of the biological weapons version of Lyme disease, they deployed it against U.S. civilians in Lyme, Connecticut. Theory number two. The United States military either intentionally or accidentally deployed the biological weapon version of Lyme disease against civilians. Theory number three. Lyme disease is a biological weapon accidentally released from the Plum Island Animal Disease Center, which is known to have reserves of ticks and whose bacterial research focuses include Babasiasis and Rickettsia. These theories and this presentation so far is contained in a book by Chris Newby from 2019 called Bitten: The Secret History of Lyme Disease and Biological Weapons. She interviews Burgdorfer himself and a few other people who might have been involved in such a program. And she advances the theories that I just demonstrated. So let's consider these three theories, the pros and cons. The first theory, that the Russians did it. The evidence in Newby's book is a few comments by Bergdorfer in the interviews where he makes vague references to Russians. And there's also some people claiming that some of the biological weapon specimens were stolen by the Russians. In my opinion, what Bergdorfer was saying is more like saying that biological weapon escalation was a necessity because of the Russians being viewed as a threat. He wasn't literally saying that individual Russians were involved in the biological weapons program. Other cons for this theory: it's generally absurd. The Soviet archives were opened up in 1991, so shouldn't this be confirmed if it were true? I mean, if the Russians intentionally attacked civilians with this weapon that they stole, I mean, why? I mean, that would be a major crime, and what would they benefit? The interviews conducted by Newby were made difficult by Bergdorfer's condition of Parkinson's. The second theory, that it was intentionally or accidentally released by the US military. This would have been something that they had trained for. For example, Operation Big Itch would have been nearly identical to what would have happened in Connecticut. In addition, this theory this would explain why the source of the Lyme disease outbreak had access to that particular species of bacteria, because if it was a biological weapon, then the US military would have had access to it. But for the same problems with this theory are present in the theory that the Russians did it. I mean, it was a major crime with little profit. So why would they have done this? If it was supposedly a scientific experiment in order to see the effect it had on a civilian population, why would it need to be an actual biological weapon instead of just releasing ticks with isotopes like before? Or another thing that happens frequently is the use of a less harmful disease that's contagious in a similar respect, so that you can release the less harmful disease as a way of observing what would have happened with a real biological weapon. And why wouldn't they have done that if it was a scientific experiment? The third theory is that it's accidentally released from Plum Island. This is the most popular theory. There have been a lot of people alleging this. The theory goes that they were researching Lyme disease as a biological weapon at Plum Island, and that it was accidentally released. Well, it wouldn't have been the first time a biological weapon was accidentally released. And since Lyme disease and the ticks that carry it were, I mean, to the extent that Lyme disease before the 72 outbreak is the same as it is now, it was already endemic to the region to the northeast, which is where Plum Island was. In fact, Plum Island was essentially in the direct center of the 72 outbreak. And Plum Island Research Center was also known to be conducting biological weapons work. This was denied by the government until recently, when it was proven that they actually were. On the other hand, there is no literal evidence that a biological weapon based on Lyme disease was being researched at Plum Island. Although they were studying rickettsias and tick-based illnesses and ticks themselves, we don't have evidence that the exact manifestation of Lyme disease in 72 was present at Plum Island. In addition, the accidental release of one particular disease would have been a one-off event. So you could imagine that Plum Island accidentally released a disease which became the 72 outbreak, but what about the other outbreaks of tick-borne illnesses between 68 and 75? I mean, it wasn't only Lyme disease. The usage of Lyme disease as a biological weapon, in the way I described, requires Borrelia burgdorferi and a microbe that masks those antigens. So in Newby's book, this is the theory that's presented, that these two microbes came together as a biological weapon from what was before just two separate illnesses. Rickettsia helvetica traveled from Switzerland and was developed by Fort Detrick, and Borrelia burgdorferi was taken from nature, and the combination of the two supposedly is a powerful biological weapon that evades an immune system. Well, the Swiss agent does exist. It was discovered by Bergdorfer. Does it mask the antigens of Lyme disease? This isn't a known fact. I mean, Lyme disease is indeed hard to detect, but it isn't known that there is any particular agent that masks its antigen. Is the Swiss agent closely related to a rickettsia present in Lyme disease patient's sera? Well, this also is not specifically known. I mean, there are many rickettsias that occur in many different combinations in many different ticks. Far more than even the diseases caused by rickettsias, because it's just a bacteria. So if you want to identify the 72 outbreak of Lyme disease as a biological weapon based on a Rickettsia helvetica variant, well, you would have to demonstrate that such a variant is present there, and also that it's not present anywhere else. Newby attempts to do that research, which is easier now than it was in the 70s and 80s because of genome sequencing. But what Newby does not identify is a specific Rickettsia bacteria present in post-72 Lyme disease sera. There are a few different Rickettsias associated with Lyme disease, but if you wanted to, if you seriously wanted to identify the American version of Swiss agent, you'd have to have like one particular Rickettsia that's present in Lyme disease patient sera that wasn't present before 1968 and is present in the outbreak but may or may not be present in ordinary infections of Rickettsia burgdorferi. So at the genus level, no part of this evidence is better than circumstantial. Rickettsia as a genus is present in the Lyme disease outbreak and is also part of biological weapons research. You could say a similar thing about Borrelia, but direct evidence at the species level for a particular species being researched as a biological weapon and also being a core component of the Lyme disease outbreak, that's not present. So basically all of this evidence is circumstantial at the genus level. The species information in this story is, for example, Willy Burgdorfer worked on biological weapons and went on to discover Borrelia burgdorferi, and in the book the Swiss agent is presented as like a mysterious bacteria that Willy Burgdorfer discovered and then immediately silenced when it became politically convenient to champion Borrelia burgdorferi as the only cause of Lyme disease. But the Swiss agent in its connection to Lyme disease today is not something, isn't there's no specific evidence for that yet. Consider this reasoning. Any disease capable of manifesting in an outbreak that appears like a biological weapon would have been investigated by any intelligent biological weapons researcher as a potential biological weapon. And Fort Detrick had a lot of money, and they had a lot of enthusiasm as part of the Cold War. So they were furiously looking through every disease they could find and attempting to identify, can we use this as a biological weapon? Can we evolve this into something that could be used as a biological weapon? They were doing this far more than the amount of times that biological weapons were actually deployed or accidentally released. So any disease that appears like a biological weapon will have one of two possible histories. Either it is a biological weapon developed in a lab that was accidentally or intentionally released, or it developed naturally and the outbreak of it would be natural, and then separately, because of its potential for use as a biological weapon, it would have been researched as a biological weapon even though it was natural. So if you want to claim that something that appears like a biological weapon is a biological weapon, you need to have some method of distinguishing between these two histories. One of these is true and one of them is false. Since we only have evidence at the genus level, we don't specifically have proof that one of these histories was actual and the other was false. And what might be considered evidence is the fact that the first Lyme disease outbreak was in the immediate years following Fort Detrick's capability of producing such a weapon. But from the standpoint of considering biological weapons in general, the fact that one of them occurred in an outbreak around the time it was being researched isn't itself evidence that it was a biological weapon, because almost everything relevant would have been an active topic of research. So without any direct evidence, what we can consider is statistics. We can apply Bayes' law, which says that the probability of A given B scaled to the probability of B is equal to the probability of B given A scaled to the probability of A. So let's say A is the event of the particular combination of bacteria species associated with the 72 outbreak and Lyme disease after 1968. And let's say B is the event where an outbreak of that particular combination of bacteria occurs in the period after such a weapon, after the production capabilities of such a weapon were made available to Fort Detrick, which is 1968, and up until the period after the dissolution of the biological weapons program in 1969. Bayes' law says that the probability that it was a biological weapon, given that there was an outbreak, times the probability that there was an outbreak is equal to the probability of an outbreak given that it was developed as a biological weapon times the probability that it was developed as a biological weapon whatsoever. Now, if it were to occur naturally, there would have been an outbreak. It's not going to occur naturally and then not infect people. So the probability of an outbreak occurring is equal to the likelihood that it was developed as a biological weapon and then went on to be released, plus the probability that it was something that evolved naturally in the same time period. Which means that overall the likelihood that Lyme disease after 1968 was originally developed as a biological weapon is equal to the ratio between the likelihood that this weapon was developed and then went on to be released to that the likelihood that it was a weapon that was released plus the likelihood of it being natural. Without a reason to suspect that it can't evolve naturally, which is not something I see any reason to suspect. What we're comparing is the likelihood that the mutations from Rickettsia helvetica that produce an antigen masking agent occur in nature or occur in the lab, except for it to occur in the lab, we would additionally have to suppose the unlikely event that it was released. So what was occurring in labs was the breeding of ticks and tick-based illnesses for their best effects. So they were intentionally trying to breed a culture of bacteria that had these effects. So on the one hand, we're considering the likelihood that the random mutations in the genome of the Swiss agent produce the biological weapon that we're supposing is behind the 72 outbreak. And on the other hand, we are considering the likelihood that random mutations in nature produce this exact same antigen masking agent. Without genome editing, we're just asking, are random mutations in a lab population of ticks more or less likely to produce the antigen masking agent than random mutations in nature? Well, there's more ticks in nature. In a lab you would have to surmount that entire random process and then go on to have it accidentally be released. Whereas nature just has huge populations of ticks and livestock, all, chaotically breeding against each other. In summation, circumstantial evidence at best allows us to produce a probability argument like this, and if you want to do better than that, you would need to identify You need to prove the existence of the antigen masking agent in Lyme disease, and then figure out where it came from.